What a great time to be learning about viral and prokaryotic genomes. We'll talk a little bit about COVID-19 genome, but to start, let's look at this graph again. I had introduced this earlier. This is a graph where on one axis we're putting the size of the genome, and then we're putting the amount of coding DNA. And so we use this to illustrate the C-value paradox, that once you get up here into the eukaryotes, that there's no correlation at all between the size of their genome and how many genes they have or how much of the genome is non-coding. So it kind of flattens out. But notice among the viruses, right, which are the blue, and among the prokaryotes, which are kind of here in these brownish and yellow brown colors, and even a little bit among the unicellular eukaryotes, there's a 1-to-1 ratio. And so basically, as genomes get bigger, the number of genes get bigger, and this is because they have very, very little non-coding DNA. So in this category, the prokaryotes, bacteria and archaea, are more similar to the viruses in their genomes. They have smaller genomes, not as small as most viruses, although there is some overlap, but they all have very, very little non-coding DNA. So that's the main difference between eukaryotic genomes and the genomes of prokaryotes and viruses. So let's take a look at viral genomes. Viruses are little bits of genetic material that is encapsulated in a outer protein coat. And sometimes they're membranes, but often not. It's just a protein coat that provides a stable environment for that little bit of genetic material. And one of the most interesting things about viruses is they can have very, very diverse genetic material. They can be made of RNA, like the COVID virus. They can be made of DNA. That RNA can be single-stranded. The COVID virus is a single-stranded RNA virus. They can be made of double-stranded RNA, which is less common, but they can be made of double-stranded DNA, so just a huge variety. All prokaryotic genomes, all eukaryotic genomes are simply made of double-stranded DNA. So viruses have a wider range of genetic material that can be the basic component of their genome. And as we mentioned, they have very, very little non-coding DNA. In fact, many viruses take this to an extreme level where they have a very, very efficient, compact genome. So we'll look at an illustration, and then we'll actually look at a map, a simple map of the COVID virus genome. So why do they have such a small genome? Well, basically, it's for efficiency. Viruses are a sneak attack. They go in, they need to make lots and lots of copies for themselves, and so any little bit of advantage they can gain in how fast they can do that or how many copies they can make is an advantage for them. And so they're kind of stripped down for efficiency. The fewest genes necessary to just be able to get into the next cell, make a copy of what they need to replicate themselves, and start over again. And viruses can really, really limit the number of genes they need because they are parasites. So they don't need to maintain homeostasis. They don't need to gain energy. They do all this by hijacking it from their hosts. So the small genome is an adaptive trait for this kind of hyper-parasitic lifestyle. And in fact, their genomes are so small, there's a diagram here. I'm going to make it a little bit larger just so you can see it. This is using English words to illustrate the principle, and it's not really great, Shakespeare here, but it illustrates the point. So imagine that this is a viral genome down here in the bottom, and each of these subsections represents a different gene. Now notice that some of the genes overlap, and this is not in all cases, but many viruses do this. So this word adds is used in sentence number 1, but it can be reused in sentence number 2. And rather than writing it twice, you can overlap. And in some cases, viruses even have genes that are in different reading sequences, but that cover the same general area. And again, I don't know, if you guys can think of a better sentence than this, great, that maybe is bonus points. But these are not really great sentences, but they make sense. One who Tad saw, notice, is just the same letters, but out of reading frame from these other sentences. So we can have genes that overlap and even genes that are out of reading frame. So let's take a quick look at the COVID-19 genome. So I've looked on the NCBI website. I've got it here. You guys can take a look at it if you're interested. It takes a little bit of practice to learn how to navigate this, but it's kind of interesting. So because this is a new virus, there's still some things to figure out, but it's going to be similar to other coronaviruses that are better known. So there's a polyprotein here, which it says location here is from 266 to 21,500. So this spans, about 21,000 base pairs. A polyprotein is a long transcript that many different proteins can be made off of. So typically there are a half dozen or more proteins that will be made from these, and these have very specific properties. They might help the virus to hijack the mechanisms of their host, or they may code for a part of the protein code. In this case, it looks like this is actually part of the glycoprotein, so part of that protein code on the outside is made by this gene here that they've designated S. And then there are open reading frames that are unlabeled, so these may be new ones that they need to look at, and then they can compare them. Here's another part of the nucleocapsid phosphoprotein. So these ORFs are simply open reading frames that they've identified, look like they code for protein, but they're not sure exactly what it is. There's some information about protein ID, and you could blast search it or look for similarities. So there are dozens and dozens of research labs now that are very quickly and with lots of people looking at this. But notice there's very little non-coding DNA. So the polyprotein here, from the part they've designated 0, it looks like there are 266 RNA nucleotides before that first polyprotein begins. It ends at 2155, and this next one starts at 21563. So there's only a dozen or so base pairs in between these two proteins. So very, very little non-coding DNA. No introns, no transposable elements. Cut down, maximum efficiency. And that's the way nearly all viral genomes are. Okay? So on to prokaryotic genomes. Prokaryotic genomes are much more similar to viral genomes than they are to eukaryotic genomes. They are efficient. They are almost certainly the ancestral genome type for all life on Earth. Now remember, viruses may have come from a broken bacteria or a bacteria that evolved slowly over many steps to be hyper efficient. So this may even be the ancestral genome type for viruses or some types of viruses. But certainly it's the ancestral genome type for all life on Earth. It is circular, so their genome is in one large piece. Here's a map of the E. coli genome with all of the different features mapped onto it. And notice it's much larger than most viral genomes, although there are some viral genomes that get fairly large. So this is on the order of about 6 million base pairs for the E. coli genome, so 6 megabases. And we have got lots of different parts that are mapped onto this genome. Okay. Now, almost all prokaryotes, bacteria and archaea, have one large circular genome. Remember, double-stranded DNA. That's the only type of genome that living organisms have. Now, in addition to their main genome, they can have a plasmid. And a plasmid is like, think of it like a little bonus chromosome. They're also circular. Some of them can have 2. Most only have 1. But they're not necessary for survival. But they can provide additional bonus features for the organism. And we take advantage of these plasmids when we transform bacteria to carry other genes. So, for instance, we have bacteria that have been transformed with human insulin genes, so they'll just make human insulin for us. We do this to clone genes. I do this in my lab quite regularly. In fact, I'm doing some of that this week for target genes that I'm interested in studying. And although it's not quite as extreme as viruses, these prokaryotes do have 85 to 95% of their DNA being coding DNA, so very, very little non-coding DNA. This, by the way, here is an artificially colored micrograph, a scanning electron micrograph of a genome of a E. coli. So you can see it's that circular, kind of like a rubber band, but made up of double-stranded DNA. And although it's rare, some prokaryotes can have introns and also transposable elements. So although introns and transposable elements are a very important feature for eukaryotes, they're a defining feature of eukaryotes. They actually evolved before eukaryotes were around. but they're not really important. They evolved in the prokaryotes, but did not become an important defining feature of genomes until the eukaryotes really started to get going. Okay? Now, because of this little non-coding DNA, there's a very strong correlation. It's not quite one-to-one. Notice there's a bit of a spread here at the end. Amongst the archaea, it's a little bit tighter, but among the bacteria, there is some range. So there are some bacteria that are a little bit of rule breakers and have a little bit more non-coding DNA. And so we get, especially at the larger end here, a little bit of a spread. But it's not anywhere near like the plateau feature like we saw in the eukaryotes. And there are even some predictions that you can make based on the lifestyle of the bacteria or the archaea. If it's free living, there's just a wide range. Some are really large, some are really small with a little more, a little less non-coding DNA. If they're parasites, they're going to be on the small side. And if they're symbionts, and this is important, remember when we talked about the origin of the chloroplast and the mitochondria? Those started as obligate symbionts. Now, obligate means that they can only survive inside some sort of a host. Symbiont means they're living there, probably in either a commensal, meaning they're not harming or benefiting their host, or maybe even in a mutualistic, which means that they're benefiting their host, a mutualistic relationship. And so those have very, very small genomes because they are often adapted for efficiency. And also they don't need as many genes because the host can perform many of the physiological functions that living organisms need to perform. This is just a diagram reminding us of these plasmids. Sometimes they can be transmitted by prophages, so they can be transmitted from one bacteria to another. Sometimes they can have virulence factors on them. So for instance, there's been documented cases of resistance to antibiotics or other virulence factors that can be transmitted from one bacterium to another on these plasmids. And so they can cause problems for their hosts, but also they're incredibly useful. We use these, we artificially make our own plasmids and then transform bacteria by inserting those plasmids into a new bacterium, and then that bacterium carries whatever features are on that plasmid. I always think of this, imagine that you were able to look at an individual that had a genetic feature that you liked. Say you saw somebody walking down the street and said, wow, you've got beautiful, thick, curly hair. I wish I had hair like that. And they said, oh, well, here, borrow some of mine. And a little tube would come out of them and stick into you, and you could transfer that little bit of genetic material. Now, we're not quite, obviously humans can't do that, and we're not even quite to that stage technologically, but we can do some things and transfer these. But bacteria have been doing this for billions and billions of years. They do it naturally in some types of bacteria, and we take advantage of it and do it artificially when we want to add a little bit of genetic material to a bacteria. So there are three methods by which this can occur. In general, this is called horizontal gene transfer. Okay, so you just need to know that, and it's a really, really important factor and feature in the bacteria and the archaea. This occurs occasionally when a mistake is made by a virus. A bacteriophage is a virus that attacks bacteria. And normally they insert their own genome into the bacterium, they hijack the cell, they often will kill the cell through the lytic cycle, make a whole bunch of new bacteria, new viruses. But every once in a while, one of those viruses will carry a little bit of the host DNA. It's like a mistake. And rather than incorporating the viral genome into its little protein capsule, it incorporates host genome. And so if this gets injected into the host, it can either as a plasmid or being incorporated into the host genome can now be a new bit of genetic material that this bacterium did not carry before. So that happens naturally, but basically through a mistake in the way the viruses are packaged. We use transformation when we do this artificially, but this can happen in nature also. If a little bit of free-floating DNA gets into the cell and gets incorporated in the genome or becomes a plasmid, that would be a transformation. We do this artificially by heat shocking or by electroshocking the bacteria after we mix them with artificially constructed plasmids, and so they carry that new DNA. And some bacteria are much better at doing this and have more ability to uptake a plasmid or just have plasmids in general, so they're useful. E. coli is actually really, really useful and used quite frequently for transformation reactions. And then the final one you need to know is a type of horizontal gene transfer called conjugation. And that's like the one I was talking about. If humans could transfer DNA from one to another through a tube, that would be like conjugation. But many bacteria do this naturally. And in fact, oftentimes the ability to transform other bacteria is carried along with this plasmid. So one bacteria will shoot out a little tube called a pilus, or pili for plural, and then it will transfer a copy of that plasmid into the new bacterium, and now that new bacterium carries that, whatever that is. So if that is a resistance to antibiotic, we now have two bacterium, and maybe not even in the same species, but two bacteria that are now resistant to whatever antibiotic. Okay, so horizontal gene transfer plays a massive role amongst the prokaryotes. It's present in eukaryotes, but does not play as critical and as major a role. So, as we've been able to identify, sequence, and map these different genomes, we can ask more complex questions beyond just how big is your genome, right? We could ask things like, hey, are the genes on your genome in the same order as the genes in a chicken's genome? You know, you might have many of the same genes, but are they arranged differently? And we'll look at that in eukaryotes in the next unit, but let's look at it in prokaryotes. So here are two very closely related bacteria, just two strains of E. coli. If their genes were in the exact same order from the beginning of their genome to the end of the genome, we would see a very simple horizontal line. Green basically represents genes that are in about the same place, but we'd see a horizontal line with no breaks and no random genes in different places. But notice some of them have been shuffled to new spots. One large chunk has been completely reversed, so they're still in relative order, but backwards to forwards to backwards instead of backwards to forwards, right? So that represents one of these inversion events that we talked about when we talked about mutations. So each of these translocations of genes and inversion events represents a mutation. And so notice these are very frequent. Even closely related strands have many, many instances of a movement of a gene. And if we take two bacteria that are somewhat distantly related, then we say every dot represents a gene they share in common, but notice that there's really no pattern at all in the order of those genes. And so this tells us that amongst the prokaryotes, that genome rearrangement is incredibly frequent, happens all the time. And so much so that even after short periods of separation between diverged species, even though they may have 90% of the genes still the same, those genes are completely rearranged in a different order. And so by and large, it's not 100%, but most of the time, it does not matter where a gene is located in your genome. As long as it's there and you can use it when you need it, you're fine. So there's no really set order to the vast majority of the instruction manual for prokaryotes. We'll look at eukaryotes in the next unit. Okay, so just realize that. Now, gene order, I'm going to put another vocabulary word up here, which we'll also do when we do the eukaryote gene order. But this is also called synteny. So I'll put that in parentheses here. It's a good vocabulary word. You should know it. Synteny is simply the order of genes on the genome. And so realize that there are very low levels of synteny, even between marginally closely related bacteria. There's going to be a very low level of similar order of the genes. Another thing that we've been able to identify and look at are patterns in the way that organisms are using their genomes. The first one we're going to discuss is codon usage bias. Now this is a nice intuitive name. It means exactly what it sounds like. So think about this. We talked about how the genetic code was degenerate. Remember, like proline, for instance, is coded by CCC, also CCU, CCG, CCA, all of the above code for proline. And so it shouldn't matter because that is a synonymous mutation. When a random mutation happens and changes a CCC to a CCA, for instance, it shouldn't matter. And so over time, as all these random mutations build up, we would expect to see roughly equal proportions of codons in the genome. Like 25% would be CCC and 25% would be CCG and so on. However, when we actually look amongst the prokaryotes, we do see that there is bias. And so at first we didn't know exactly why this might be. If it's a completely synonymous mutation and we're having random mutations occurring, throughout the history of organisms, why wouldn't we see roughly 25% of all the possibilities? And people began to figure this out when they noticed that if there was a bias in the codons, there was often also a bias in the tRNAs that brought in the amino acids as we were building these protein chains. And so that got them thinking, oh, maybe This is something that helps with the accuracy of DNA replication, but even more so, there is some replication and transcription, but even more importantly, and in fact, I'm going to put that here. Oops, sorry, I'm going to put that here under this last one. This greatly helps with the translation, so the accuracy, or maybe even better term, efficiency of translation. And so here's how this works. Imagine you are building something out of Legos and you're trying to do it very, very quickly. And every time you need a certain Lego to build in and put in, there are 4 different boxes you can draw from. And so maybe it's only a slight hesitation, but you hesitate because you maybe want to use them equally or you don't want to run out. So you try to draw equally from those 4 different boxes. Now you're like, well, why don't you just put all those same Legos in one box, and then you don't have that hesitation. And so basically that's what a codon usage bias is it's an adaptation for efficiency of copying and especially translating these genes and making the proteins that they need. So if you have mostly CCC instead of CCA, CCG, that makes it a little bit more efficient. So where efficiency is important, natural selection has worked to create these biases. So a bias is a natural selection response for an increased efficiency of transcribing and replicating a little bit, but even more so translating your genes into what they need to do. Now, this is not important for all genes, but when it is, there is a bias there. Okay? The next type of bias we're going to talk about is GC content bias. Now, GC content bias, again, very intuitive name. Basically, we count up all the G's and C's in a genome, and then we count up all the A's and T's. Now, hopefully it's obvious to you why we don't count up all the G's and then count up all the C's. Right? So think about it. That would just be redundant because all living organisms have double-stranded DNA, so every time you've got a G, you also have a C. So we just count those up and then we compare them to the AT pairs. And again, we predicted ahead of time that it really shouldn't matter. Random mutations, you can have some G's, some C's, you can have some A's and some T's. Shouldn't matter. And so we should see roughly 50%. Yet when we look, we do see some significant bias towards having more G's and C's in some organisms and more A's and T's in others. So here's a lineup. This includes eukaryotic organisms, which we'll talk about in the next unit also. But notice amongst the bacteria up here. The Mycobacterium, is that the only one we have up there? Yeah, that's the only bacteria we have here. But some bacteria have as much as 80% G's and C's, whereas other bacteria have as little as 25% G's and T's. And so again, for a long time, people didn't understand why this would be. We would predict at the outset that it should be roughly 50-50, not that significantly different. There shouldn't be an advantage to having more G's and C's. And so people begin to try and figure out this. And the explanations for this are varied and are different for different organisms. But there are 2 general patterns that we see. We see mechanistic explanations, which means maybe there's just some mutational bias, right? A mechanism of mutation that causes G's and C's to occur more frequently when mutations occur. Or maybe there's a bias in the proofreading mechanisms towards one or the other. So that would cause a buildup and really wouldn't be any for an advantage. But there actually might be some advantages to this in certain circumstances. So I'm going to tell you one example of where there might be a selectional reason for why we might have more G's and C's. And it comes down to the molecular characteristics of a GC pair versus an AT pair. And if you think back, maybe you don't remember this, and that's fine. But if you do, you think back, a GC pair is different than an AT pair. Obviously, there's some differences at what atoms and how the atoms are arranged, but even more importantly, when they pair up via hydrogen bonding. And GC pairs have 3 hydrogen bonds, where AT pairs only have 1. So they're slightly more stable. So if you live in an environment where stability of DNA is important, you want a little bit more of a stable DNA helix, having more G's and C's is an advantage. And so natural selection would bias you towards mutations when they occur, even if they're occurring randomly, the GC mutations would be kept more commonly than the AT mutations. And so we'd end up building up over many, many generations more G's and C's. If on the other hand, you lived in an environment where you wanted to be able to unzip your DNA a little bit more easily, you wanted it a little less stable, then there would be a bias the other way. And so that is one example of a selectional reason why we might have a bias in GC content. So know the definition of GC content, know that there might be some mechanistic explanations like types of mutations or proofreading mechanisms, but that there are often also selectional reasons for more stability. Okay? And then lastly, this is really important, number 2 here. This can help us identify, and I'm even going to modify this. Let's put in recent HGT genes that have been transferred over via HGT. So remember, HGT is an acronym for horizontal gene transfer. Okay? So horizontal gene transfer plays a very, very important role, and we've been able to identify this. So let me talk you through just basically how we identify this. Okay? Amongst the bacterium archaea, whatever selectional or mechanistic things are causing you to have a certain GC content, that's genome-wide. So basically, if we look across in a bacteria genome, we look at the first part, the middle part, the end. It goes in a circle, remember, though. But we look across the whole genome, and you're going to see roughly the same level of GC content because all those processes are genome-wide. And so if we find an area of the genome where, hey, look, the GC content here is unusual, it's different than the rest of the genome, that is a good indication that that has been a recent horizontal gene transfer event from a bacteria with a different GC content level. Now, the reason I added recent to this is over time, the selectional processes, the mechanistic processes that are acting on that bacteria will work to bring the GC content of that newly transferred portion of the genome to bring it to whatever level the bacteria, the rest of the genome is at. But if it's recent enough, then we might be able to identify significantly different. Say, hey, look, there's a horizontal gene transfer event. So we find that difference in GC content. It's a good indication of a horizontal gene transfer event. And as we've looked across many, many bacterial and archaeal genomes, we found that horizontal gene transfer events occur all the time. They're very, very common in bacteria, so much so that we really can't do meaningful standard phylogenies for bacteria. So remember, we talked about this, right? When we talked about this bifurcating, tree-like pattern that we see in phylogenies, we said that there are 2 places where we don't see a tree-like pattern. Number 1 is within a sexually reproducing species. So if I take a whole bunch of humans and look at their patterns, they're going to make a network, maybe close if they're inbred, or very distantly if they have been not breeding closely within a single population. But no matter how we look at it, there's going to be some level of that network-like pattern. And then the second place we talked about it was amongst species of bacteria. And the explanation is different. So the reason that we see network-like patterns and not phylogeny or tree-like patterns amongst the bacteria is because of horizontal gene transfer. Okay, so it plays a really critical role amongst the prokaryotes. It is present in eukaryotes, but plays a much, much more minor role. All right, that wraps up our virus and prokaryotic genomes.